I'm Tom Sexton. I'm a graduate student in ASIP's Cousins Lab. My research focuses on the photosynthetic response to drought. And specifically, my work has been on wheat, especially here in the Palouse. When we think about the challenges that wheat is presented with, low water availability, especially in the summer when it's hot and dry, is one of the major challenges that wheat faces as it continues to grow and attempt to be productive and set seed. And so my research is really focusing how under these low water stressful conditions, we're able to maintain productivity in crop plants. And so the way that we do this is through a combination of physiological measurements and imaging. And so we can directly measure photosynthetic and transpiration rates using controlled chambers that clamp onto a leaf. And we combine these physiological measurements with visible and thermal imaging that provide us the unique opportunity to quantify plant performance non-destructively. And so visible imaging can quantify whole canopy traits and whole plant productivity, while thermal imaging measures leaf temperature, which is a really important trait because staying cool and avoiding overheating under drought conditions where it's hot and dry is a really important trait. And so these tools allow us to distinguish which mechanisms and physiological differences account for greater performance in plants that are able to continue to fix carbon and grow as environmental conditions are harsh and water is limited. And so the ultimate goal of this research is to someday understand the mechanisms that plants use to avoid being damaged by reduced water availability well enough so that breeders and geneticists in the future can stack beneficial traits together from different varieties in order to ensure that future crops are able to tolerate future conditions of increased stress in order to continue to produce the food we depend on.